Now we are going to see the difference between mitosis and meiosis. So here in mitosis, it occurs in the stomatic cell, and here it occurs in the reproductive cells. Okay, the mitosis will take place in the stomatic cell, that is body cell. Here in the meiosis will take place in the reproductive cells like gametes, okay, like gametes, that is it might be the sperm cell or in the egg cells. Then here in mitosis, it is involved in the growth and occur continuously throughout the life. Mitosis is involved in the growth and it occurs continuously throughout the life. Here in meiosis, it is involved in gamete formation only. Meiosis will be involved in the gamete formation only during the reproductive active age. Okay, well, only during the reproduction time the meiosis will take place in the gamete cells. Here in the mitosis, it consists of single division. In mitosis, the single division only will take place. In meiosis, it consists of two divisions. So the meiosis will have two divisions. In mitosis, the two diploid daughter cells are formed. In mitosis, two daughter cells are formed. Here in meiosis, four haploid daughter cells are formed. Once again, here in the meiosis, two daughter cells, okay, two diploid daughter cells are formed. Here, four haploid daughter cells are formed. Here, the chromosome number, okay, the chromosome number in the daughter cell is similar to the parent cell. So, it will be seen in the same number of chromosomes will be seen in the daughter cell also. Okay, here in the meiosis, the chromosomes, okay, the chromosome number in the daughter cell is just half the parent cell. Okay, so it will be seen in the haploid in nature. Okay, so the chromosome number in the daughter cell is just half of the parent cell. In mitosis, identical daughter cells are formed. In mitosis, identical daughter cells are formed. In meiosis, daughter cells are not similar to the parent cell and are randomly assorted. Here the daughter cells are not similar to the parent cell and are randomly assorted. Today we are going to see about organization of tissues. In that we are going to see about meiosis. The term meiosis was coined by Farmer in 1905. It is the kind of cell division that produces the sex cells or the gametes. It is also called reduction division because the chromosome number is reduced to haploid from diploid. Meiosis produces four daughter cells from a parent cell. Meiosis consists of two divisions. They are heterotypic division or first meiotic division, homotypic division or second meiotic division. Heterotypic division. It divides the diploid cells into two haploid cells. The daughter cells resulting from this divisions are different from the parent cell in the chromosome number. This consists of 5 stages: Prophase I, Metaphase I, Anaphase I, Telophase I, Cytokinesis. Prophase I. Prophase I takes a longer duration and is subdivided into 5 stages. They are leptotin, zygotin, pachytotin, diplotin, and diakinesis. Leptotene. The chromosomes become uncoiled and assume long thread-like structure and takes up a specific orientation inside the nucleus. They form a bouquet stage. Zygotin. Zygon means adjoining. Two homologous chromosomes approach each other and begin to pair. Pairing of homologous chromosome is called as synapsis. Paciotin. Pacus means thick. The chromosomes are visible as long paired twisted threads. The pairs so formed are called bivalents. Each bivalence now contains 4 chromatids. It is also said to be tetrad stage. Homologous chromosomes of each pair begin to separate. They do not completely separate but remain attached together at one or more points by X-shaped arrangements known as chiasmata. The chromatids break at these points and the broken segments may be interchanged or otherwise said as crossing over. As a result, genetic recombination take place. Diplotin. Each individual chromosome of each bivalent begins to split longitudinally into 2 similar chromatids. The homologous chromosomes repel each other and separate. Chiasmata begins to move along the length of the chromosome from the centromere towards the end, resulting in terminalization. Diakinesis. The paired chromosomes are shortened and thickened. The nuclear membrane and nucleolus begin to disappear. Spindle fibers make their appearance. Metaphase. Metaphase I. The chromosomes move towards the equator and finally they orient themselves on the equator. The 2 chromatids of each chromosomes do not separate. The centromere does not divide. Anaphase I. Each homologous chromosome with its 2 chromatids and undivided centromere move towards the opposite pole of the cells. This stage of the chromosome is called dyad. Telophase I. The haploid number of chromosome after reaching their respective poles become uncoiled and elongated. The nuclear membrane and the nucleolus reappear and thus 2 daughter nuclei are formed. Cytokinesis I. The cytoplasmic division occurs and 2 haploid cells are formed. Homotypic division. In this division, the 2 haploid cells formed during the first meiotic division divide into 4 haploid cells. The daughter cells are similar to the parent cell in the chromosome number. It consists of 5 stages: Prophase II, Metaphase II, Anaphase II, Telophase II, Cytokinesis II. Prophase II. The centriole divides into two. Each one moves to the opposite pole. Asters and spindle fibers appear. Nuclear membrane and nucleolus disappear. Metaphase II. The chromosomes get arranged on the equator. Two chromatids are separated. Anaphase II. The separated chromatids become daughter chromosomes and move to opposite poles. Telophase II. The daughter chromosomes are centered. The nuclear membrane and a nucleolus appear. Cytokinesis II. Two cells are formed from each haploid daughter cells, resulting in the formation of 4 cells with haploid number of chromosomes. Significance of meiosis. The constant number of chromosomes in a given species is maintained by meiotic division.